This is the second video for cell division. In this video, we will discuss meiosis and how it compares to mitosis. Meiotic cell division is specifically used in sexually replicating organisms to produce the sex cells, or gametes, necessary for sexual reproduction. Meiosis involves two rounds of cell division called meiosis I and meiosis II, but only one round of interphase. Thus, the resulting daughter cells are different than in mitosis. In meiosis, 4 daughter cells are produced, which are haploid. They contain only one set of the genome and are genetically unique or different than the parent cell. For example, in humans, a cell which would contain 46 chromosomes would undergo meiosis to form egg or sperm, depending on whether that cell was in the ovaries or in the testes. The egg or sperm are both haploid, thus they would contain 23 chromosomes. These haploid gametes then come together upon fertilization, each bringing their own genome and generating a 46 chromosome diploid cell. This fertilized egg is called the zygote. The zygote would then undergo multiple rounds of mitosis to grow and replicate and make many more cells needed for cell growth into an adult. To demonstrate meiosis, we will walk the same cell that we saw in the mitosis video through the stages of meiosis. Remember, this cell was a diploid cell with 4 chromosomes, 2 from mom and 2 from dad. Just as in mitosis, the cell must first go through interphase, including S phase, where those chromosomes will replicate to form sister chromatids. The cell will then go through the same phases that occurred in mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. However, because there are 2 rounds of cell division in meiosis, there must be 2 versions of each phase. After cytokinesis 1, the 2 daughter cells immediately enter into the second round of division and begin prophase 2. The first round of phases of meiosis have similar but not identical cellular changes occurring when compared to mitosis. The 2 phases with key differences, marked here by a star, are prophase 1 and metaphase 1. The cellular changes in the second round of meiosis are identical to those you already learned in mitosis. Before we continue to diagram this cell as it goes through meiosis, we will first discuss the unique cellular process that occurs during prophase 1 of meiosis. In prophase 1, an interaction called crossing over occurs between the non-sister chromatids of homologous chromosomes. Remember that homologous chromosomes are chromosomes with the same gene sets, but one came from mom and the other from dad, as represented here by the maternal light blue and the paternal dark blue chromosomes. The 2 copies of each homologous pair are the sister chromatids, which formed during DNA replication. As the cell enters prophase 1, the 2 homologous pairs of sister chromatids line up with each other, coming together to lie side by side, gene for gene, in a process known as synapsis. Because each homologous chromosome is a pair of sister chromatids attached to a single centromere, a pair of synapsed chromosomes creates a 4-stranded structure, 2 pairs of sister chromatids aligned along their length. The whole unit is called a bivalent or a tetrad. The bivalent will then undergo crossing over, where a piece of DNA from the non-sister chromatid exchange DNA segments. This allows homologous chromosomes of maternal origin and paternal origin to swap sequences of genes. The location of the crossing over event is called a chiasma. The final product of crossing over are recombinant chromatids. This process is beneficial because it increases the genetic variation in the offspring. For example, pretend the genes for eye color and hair color are found on these homologous chromosomes. The eye color being here and hair color being here. However, the maternal chromosome has the gene for brown eyes and brown hair, while the paternal chromosome has a gene for blue eyes and blonde hair. After crossing over, the recombinant chromatids have now mixed up those combinations because the blue-eyed gene on Dad's piece of chromatid is now next to the brown hair color gene that had come from Mom's. This increases the possible combination of traits that will be observed in the offspring. Later in the semester, when we discuss natural selection and evolution, we will learn how important variation is to the process of natural selection. Going back to the diagram of our original cell, we can now see as we move into prophase 1 that our bivalents will form. In prometaphase 1, the mitotic spindles will begin to attach to the kinetochores, but because the homologous chromosomes have formed a bivalent, the spindles will attach to each end of the bivalent instead of this individual sister chromatids. As the mitotic spindles start pulling, we will then move into metaphase 1. It is here that the bivalents will be lined up in the middle of the cell, seen here. Attached to the mitotic spindles, coming from the centrosomes on either side of the cell. Notice the difference here between metaphase 1 of meiosis and metaphase 1 of mitosis. Both have chromosomes lined up in the middle of the cell, but the organization of those chromosomes is different. This is the key step in diagramming meiosis correctly. The bivalence will release itself as the cell moves into anaphase 1, causing the sister chromatids to be pulled to opposite sides of the cell. Notice that this is different than in mitosis because our sister chromatids are still attached together. The cell will then undergo telophase 1 and cytokinesis, forming two separate cells with a nucleus intact, each containing the sister chromatids that were pulled to the opposite sides of the cell during anaphase 1. These two daughter cells are just an intermediate step. They will both now begin a second round of cell division. The two cells from meiosis 1 will now enter prophase 2. Notice that there's not a second round of interphase. These stages will be identical to mitosis. In prophase 2, the nuclei of each cell will disappear, and the mitotic spindles will start to form. The mitotic spindles will attach to the sister chromatids during prometaphase 2 and then move them to be aligned in the middle of the cell during metaphase 2. Notice that this process occurs in both of the daughter cells from meiosis 1. The sister chromatids will then separate from each other as the cell moves into anaphase 2, as seen here. The division will complete during telophase 2 and cytokinesis, in which each one of the two cells at anaphase will be divided and nuclei will reform. As we look to see which chromosomes are now in those nuclei, you should be able to observe that each of those cells now contains half the number of chromosomes of the original parental cell. Each cell contains 2 chromosomes, making them haploid. Additionally, note that we end with 4 cells instead of 2 as in mitosis. You should notice which chromosomes are in each cell. It is not just important to have half the amount of DNA, but you must also have a full set of the genome. You do not want a cell with the maternal and paternal homologs of only 1 chromosome, because then it will be missing any information that was on the second. For example, we wouldn't want a cell with 2 of the big long chromosomes and none of the little short chromosomes. We need 1 long and 1 short in each cell. In meiotic cell division, the division of the cytoplasm differs between males and females. In females, the cytoplasm is divided very unequally in both meiotic divisions. Most of the cytoplasm remains in only 1 of the 4 daughter cells. This will develop into the functioning egg, which is called the oocyte. The smaller cells are called polar bodies, and they are non-functional and are degraded. This is in contrast to males, where the cytoplasm divides fairly equally, resulting in products that all form functional sperm. During sperm development, most of the cytoplasm is eliminated, and what remains is the nucleus in the sperm head and a flagellum to propel it towards the egg. The expulsion of the cytoplasm in sperm formation is why the egg must contain so much cytoplasm. All the cytoplasm and cellular organelles, such as mitochondria, for the fertilized zygote, must come from the egg.